very happy to be here today, and I was very happy of yesterday's also talks and participation of everyone, and I hope that today is going to be the same. With Loic, we will talk to you about clinical trials and methodology of clinical trials. So some of you might know a lot already by having being directly involved in or not, but we will maybe remind basics for those who don't have any experience of things on clinical trials. And I really think that thanks to the actuality also these days that everyone should know more about methodology to be able to review projects and everything. So today we will just do that very quickly and mentioned. We will we will review protocol together. So don't worry, it was a big document, but you will be helped by moderators with you to review that. So no worries on that. So first of all, we wanted to start with the first question. What is the first word that comes to your mind when talking about methodology of clinical trials? So, Maria?
Yes, I'm sharing now. A second. I think that can you, because I think that I have to stop sharing. Wait a second.
I have to stop sharing?
I think so. Would you mind? Thank you. And I think now, yeah, now we will create this word cloud. Okay. Now can you see my screen? Yes, I guess.
Yes.
Sorry about that.
Oh, it's nice the word we see. I'm happy that some of you talk about the quality, and patient involvement is one of the very important words. Complicated, we will see that maybe we can do something for that.
I like the hope.
Yes, yeah. Okay.
Do you want me to stop
Sounds
it?
Good. Yeah, I think people are still answering. Okay. So a bit of hope, a bit of complexity, but most of all also a protocol, of course, and the patient's involvement. That's very important. Okay.
I stop? Okay.
Yes, sure.
I'll take a screenshot and save it. Now you can share your screen, I guess.
Okay. So yeah, so thanks for sharing this this information with us. Oh, so it's a bit. So when we so let's start at the beginning. You received the video we worked on with Kids France, and you know it's a long process to develop a new drug. So from the molecule to the drug, we always start by sorry, my PowerPoint. Yeah, to find the right molecule that can have a therapeutic and a potential interest. So regarding the physiopathology, the mechanism of the disease, the researcher tries some new molecules. Then these molecules first need to be evaluated preclinically at the lab or with animal studies, you all know that. And then we can start trials in human with the phases 1 to 4. So I will just briefly go through them as you receive the video, and you know probably some of those. So phase 1 is the first administration in human that is performed on healthy volunteers. And we start here very carefully with low number of patients, maybe one patient to start and others then to test the tolerance of the drug. And we also have a look at the pharmacokinetics. So how the medicine is absorbed, diffused, metabolized, and excreted in the body. Healthy volunteers are never children, but it's not why we should not start development of medicine early in children. Okay. Oh, it's a bit slow on my computer, sorry for that. So then we go to phase 2, which where we establish the relationship between the dose and the effect of the treatment, trying to find the optimal dose of treatment, usually with a small number of patients too, a limited number of patients, which is quite homogeneous. Then comes the phase 3, where we test the efficacy of the therapeutic. So the number of patients is higher, representative of the population that will get the treatment. And at the end of this phase, a marketing authorization is asked and can be requested. So it's not finished when the marketing authorization is here. Phase 4 studies can happen, and in real life conditions usually. So epidemiologic studies on large population. We studied the rare adverse reactions that can happen and that we could not have seen maybe in early phases treatment. So among several thousand molecules, only one will make it. So it's a very long and not easy process for a molecule to get to a medicine. Development can be stopped at any phase of the development. Not everything is win in advance. In children, as in adults, it's not all about new drug development. We saw that yesterday. Other therapeutics can be used, other research can be made. Old drug used in adults can be used in children. New formulation can be proposed to adapt to children. And so depending on the available information, you can start at any phase of the development whenever you are with enough or not information for starting it at a different phase. So now I would like to talk about one important statement when we think about methodology. It's the evidence-based medicine. So what is it? Sackett told us it's the use of current best evidence in making decisions about care of individual patients by using individual clinical expertise with the best available external evidence and taking into account the specificities and the environment of the patient. So what does it mean?
Yes. Sorry, can I interrupt you for a second? I think that there's a background noise. Maybe it's from your microphone. Is it possible that's there?
Let me check. Can you hear me?
We hear you, but there's like a background noise, like, okay.
No, I have my...
Yeah, maybe it's from the microphone because it's like a...
Okay. I can try...
Yeah, if you can disconnect it. Yes.
Is it better like this? Is it like this or not?
It is better, Segolem.
Better.
Yeah.
Thanks.
Yes. So talking about evidence-based medicine and individual clinical expertise, so the role of the physician is to provide the best care to the patient. So he needs to prescribe the treatment if it's useful for him and based on evidences of the effect of the treatment. So he needs to integrate the results from randomized clinical trials into his practice. So how will he do that? So it will use the best available evidence. So we need to define a level of evidence. This level is increasing from a case study or a case report, which is the less important level of evidence, to the meta-analysis, which regroups several randomized controlled trials and which is considered as the best level of evidence. In fact, the repetition of clinical trials makes the highest level of evidence because we know that the results that are obtained by chance will not be reproducible by definition. So if we have several randomized clinical trials who say the same, we have a good evidence that it's working. So, and of course, the physician needs to take into account the patients, his wishes, the medical environment, and most of all, probably, his specificity. So we said that everything started with knowing the mechanism of a disease and a researcher who thinks that a molecule can have an effect on these mechanisms. So this is the idea of the treatment and the therapeutic hypothesis that we start with in the clinical trial protocol. So for example, in cystinosis, a very rare disease, a compound called cystine gets accumulated in lysosomes and, by the way, leads to kidney failure at the age of 10 years, probably. So that was the old hypothesis on that, the old mechanism. And then we found in the 90s a cysteamine that can link to cystine and help the cystine get out of the lysosomes. So probably that here we have an idea that cysteamine could improve kidney function and avoid kidney failure. So this is our hypothesis. But as any hypothesis, it needs to be verified. That's the principle of clinical trials. That's how we be sure that there is an efficacy and that there is no harm for the patient to participate. So we start, if you want to answer your hypothesis, stating that you have a treatment that can be efficacious, you need, for your clinical trial, an hypothesis that is issued from an adequate background. So you need to really define your disease, the treatment, why it could be useful, and how in the field of what exists and has already happened, you add something to the field and to this condition. You have one primary objective because you want to answer one question at a time, usually. So this objective should be clear, clinically relevant, important for the patient too. And to measure it, you need an endpoint, a corresponding endpoint or several endpoints to evaluate the effect of the treatment if we talk about efficacy. So this should be well defined in terms of the condition and the population. And the number of patients you will need for your study will always be calculated from this endpoint, this primary endpoint, and this hypothesis initials. You are allowed to have multiple secondary objectives as much as possible. That will be mainly exploratory unless calculations have been made on them. So usually one trial, one answer, one hypothesis, and one objective, principal objective. So if we go back to a question for you, how do you think we can demonstrate the efficacy of a therapeutic? So Maria, for the poll.
Yes, I'm sharing.
So I will read. So would it be by giving the drug to some patients?
Can you see it now in full screen or not yet?
Do you think it would be by giving the drug to some patients and observe what happens after a sufficient time? Or by giving the drug to some patients and not giving it to others and compare what happens after a sufficient time? Or by giving the drug to some patients, then not giving it to the same patients and observe after a sufficient time? Okay.
If someone of you, as always, if you are not if you cannot connect through SparkUp, you can always answer via chat.
Okay, B is for most of, okay, perfect. So we'll see B is the best answer possible.
I'll
Okay,
stop sharing.
back, yeah. So we saw that to answer our hypothesis, so we will answer the question together through all the slides that will come. So we saw that to answer our hypothesis, we needed a clinical trials that can be, a clinical trial can be seen as a tool that measures efficacy if we talk about efficacy. And this measure is always a mix of the true value of the efficacy plus some errors, because we all make errors when we do some measurements, and some other factors such as the evolution of the disease itself without treatment, other placebo effects, etc. All those factors can act and can maybe not give real evaluation of the efficacy of the treatment. So when the role of the methodology will be to get the true value of the efficacy and to get rid of these factors that annoys us to get the real efficacy and true value of efficacy. So how we can demonstrate the efficacy of the therapeutic, we need to see if patients who take the treatment do better than those who don't take it. So we need to compare, you're right. And by do better, we understand maybe that it's a treatment that is a curative treatment, or is a treatment that can be used in prevention, or it's a treatment that is maybe safer from one that exists. So different kinds of efficacy of the therapeutic. So to compare, we need what we call control groups. And often we use placebo as a control. Well, we should use it when possible and when ethically acceptable. We saw that. We don't want any loss of chance for the patients by using the placebo. So this needs to be considered very strictly. And so, but we can also use, if placebo is not possible and maybe a standard treatment exists, we can use a standard treatment. Or we can do add-on on existing treatment if we cannot leave the patient without treatment. So maybe many possibilities exist to compare, and different treatment can be used, and Loic will talk to you about this. So we have different plans that help us. Sorry for these slides. No, it's not complicated. We will just go through very quickly. To compare our drug to a placebo or another drug, we have different plans that can be used. So the most common, you know, it's the parallel groups treatment. So there are two different groups, A and B, and you follow them at the same time. One is taking the drug, the other one is not taking the drug, is taking the comparator or the placebo. Another kind of design is the crossover, when you can take into account the variability that can exist. So it can be used in different ways. Many other designs exist. I wanted to show you those ones because they are useful sometimes in rare diseases and in pediatrics. And so you need to know, we won't go through all of them, but maybe the end of one, you have one trial is one patient. So that can be very exciting, and maybe we can do meta-analysis of these clinical trials after that. So we don't need much patients. And some patients are able with some kind of drop the loser, play the winner. They have funny names sometimes, but some needs, for example, this one, the randomized withdrawal. If the patient is responder, is then randomized to the treatment. If he's not, it will be discharged. It's not needed maybe for him. So there are different plans that exist that allows you to compare. We just need to find the right one for the disease and the population that we have. So we said that comparison was important, but why? Because of the evolution of the disease. Let's take a group of patients, this one. They are very famous patients. Some are big, you see that, some are smaller. They all have the same condition. They obviously, they have an hyperactivity issue in there. We can see that. So all are different in their condition, depending maybe on their genetic, on their background, on the severity of the disease, over psychological considerations, and some known and unknown factors. So some also take potion, magic potion, we know that. So they have other treatments to do better. Some don't. So if we just give them all the treatments, what will happen? At the end of the treatment, the question is this working? If they do better, we won't know if it's because of the treatment or because of all these conditions and these differences that we have at the beginning. So to measure the treatment effect, we need to control all this. We need to compare, but maybe that's just not enough. Comparing is the first step. But does it really works? I think we're still missing something there. We need some more methodology. We should be able probably to compare comparable groups. If you take these two groups, you see that maybe one of the population seems more hyperactive than the other one. So we need a method that will provide two identical populations. So this method, it's randomization. Randomization will help us and says that belonging to a group will depend only from random and will ensure that groups, all characteristics of the patients are well distributed among the two groups. So we have comparable groups. Random is always doing a good job. And one particularity of randomization is to be unpredictable. We don't want to know in which group the patient is. So we have comparable groups thanks to the randomization, but we need to ensure that all patients, whatever the treatment they receive, are followed the same way, receive the same care, have the same examination, same advices from the doctor. We don't want to influence, we don't want really to influence the response to the treatment by acting differently from one group to another. So this is the role of the double blind. So neither the patient nor the physician knows what treatment the patient is receiving. We use a placebo for that, but it's not always possible, as we said. So if we cannot do that, we can assess the objectives and the endpoints, meaning we can measure in a single blind manner, meaning that the person who would measure the treatment effect won't know what the patient is taking. So there are other manners to manage if we cannot do the double blind. So this is great. With the methodology, we are able to ensure that we are effectively measuring the true treatment effect. And so that's the credibility of the results. We say that we avoid the bias. So randomization and double blind are methods that avoid bias in our studies. And they are among the two major methods. Other methods can exist, of course. I won't detail them. So also we need to ensure that the population we are enrolling in clinical trial is our targeted population. Is it representative of the patients who will receive the treatment in real life? So we define several eligibility criteria in our protocol on demographic data, age, sex, etc. We define the disease, if we want the disease, any patient with the disease or more complicated patients, etc. So we define also the authorized and the forbidden concomitant medications we use, and over associated maybe what we call comorbidities or over disease that would be associated with the condition studied. So this eligibility criteria are very important to define the population and be sure that will be representative of the treatment that will be given after marketing authorization. And I would say that maybe what's important here to note is that maybe when needed and when we need it, patients and children should be enrolled in clinical trials. Okay, I'm arriving at a moment that no one likes, statistics, even me. So when talking about methodology, we sure need to talk about it. So I won't be long, I swear. So we saw already the notion of chance versus real effect. I will just give you some notions, not make you experts in statistics. That would take a long time, I think. So we know that the results we are getting can have a chance to be due to random or no effect and not reflect the treatment, the true effect of the treatment. So this is why in our studies we define a probability, and you will find those elements when you review study results. So this is why I just wanted to let them know about them. So the probability, P, is the probability to observe a difference that would be only due to chance when there is no difference between the two groups. So, and our objective usually is to reject that probability. This is how we do in statistics. So we usually use a threshold to reject that, which is fixed at 5%. So usually when you look at the results, you say that if you are under this 5%, so you have a p-value of 0.05%. So you say that you have demonstrated efficacy. But if you are above it, you will say that you did not demonstrate anything. It's not statistically significant. So, and depending on this and the differences you want to show between the two groups, you will calculate the number of subjects you need in your trial. So what is the expected difference? And the smaller it is, this difference, the biggest the number of patients is. Why? Because when you want to show something which is very small, it's more difficult to demonstrate than something which is obvious, a very big difference between the groups. So if you think that there's a huge efficacy of a treatment, it's going to be easier to demonstrate. But if you think that there's an efficacy, but maybe it's not that much, depending on the comparator, it's going to be more difficult to show. So more statistics again. It's in the interpretation of the results. So you need to answer several questions when you have a p-value under 0.05. So if it's under 0.05, it's statistically significant, and it's not when it's higher. So if it's statistically significant, that doesn't make everything. Is it clinically relevant? Is it important for the patients? You need to ensure that and to interpret that for the patient himself. And is it really due to the treatment? So if you have ensured a right methodology during all your trial, you can be sure you reflect the true effect of the treatment. But if maybe you didn't do the double blind for some reason, you need to think, well, is it still okay? Am I okay? And is it really the treatment that is efficacious here? So this is all the questions we should ask ourselves when looking at results from a study. And if it's not statistically significant, well, is it because there is no difference between the groups? Or maybe we did not succeed in enrolling all the patients we wanted to, we calculated for. So we have not enough what we call power to demonstrate the difference and the efficacy. So usually we cannot conclude. We don't say that there is no difference. Maybe it's another reason why there is no difference. So that's the most difficult when we don't have significant information. But maybe we have something that can help us, even if it's not statistically significant, we can still say something and publish results that are important to have. So as a summary of what I said, when we talk about methodology, we have the content of a study protocol. So usually in a study protocol, you will find some general information. So that makes that everyone has some background information. So background, the study should be justified. Your hypothesis should be something that does not exist, that is not already answered. Then you have your objectives with your endpoints and the design you will adapt to answer your question. Then you can select your subjects. So you have all the eligibility criteria here that are noted. The study treatment, and Loic will talk about it in a moment. All the assessments and the measurements you will make during the study. So the efficacy measurements, the safety measurements, some statistics, how you will analyze your study. This is all described in a study protocol and should be before the beginning of the study. How you access the documents, the data, etc., the protection of the patients, quality control, quality assurance, how you will record the keeping, data handling, everything. You need to make sure that participation of the patients is ethic and has been sent to regulatory considerations. Financing, insurance, how you will publish, and some, of course, appendices are present in the study protocol. Maybe the scales that you will use or the documents you will use to measure everything are very important information. So that's about what's in the study protocol.